I'm Daniel Levine, and this is the Bio Report. Some 70% of potential therapeutic targets are believed to be beyond the reach of conventional small molecule therapies or biologics. Macrocyclic peptides offer a way to get at elusive targets while providing desirable characteristics of both small molecule drugs and biologics. They offer bioavailability, can permeate cells, and engage complex targets with specificity. Unnatural Products is harnessing AI to create synthetic macrocyclic peptides to pursue previously undruggable targets. We spoke to Cameron Pye, CEO and co-founder of Unnatural Products, about how these peptides can target proteins that traditional therapies can't reach, the engineering process for the company's synthetic macrocyclic peptides, and their potential to lead to new therapies for a range of conditions from cancer to obesity. Cameron, thanks for joining us.
Thanks, Danny. I appreciate the invite to be here.
We're going to talk about undruggable targets, unnatural products, and its efforts to advance a class of therapies known as macrocyclic peptides to pursue proteins that drug developers have been unable to target with conventional therapies. From time to time, we talk about undruggable targets on the show, as there have been several technologies recently emerged that promise to be able to go after these. But for the purpose of informing listeners here, what do we mean by undruggable targets?
Yeah, and this is a loaded term that I think has persisted longer than really anyone wants it to, for lack of maybe a better one. But really, what we mean by these are challenging protein targets that have beleaguered kind of traditional modalities approach to creating drugs against them. So in those traditional modalities, we typically refer to as small molecules and in antibody therapeutics or the broader biologic space. And so what typically defines these things are often their intracellular targets that are off reach to antibodies because they're not cell permeable, and they require a larger molecule to act on them because they're mediated through things like protein-protein interactions, or they're poorly structured proteins themselves. So that's, I think, kind of the canonical definition. And I'll say that we kind of think a little bit more broadly about this as well. I think particularly coming from a small molecule mindset of also viewing certain things that we currently drug with antibodies but would like to drug with small molecules as maybe falling into this kind of level 2 area of druggability as well. So I think the term is loaded, and I think what we really mean are things that don't have obvious small molecule binding sites on them and require a slightly different kind of approach to the problem.
How big a problem or opportunity do undruggable targets represent, and why should people care about this?
Yeah, well, I mean, it's, by some estimates, the kind of traditional definition of undruggability, like I mentioned there earlier, is 70% of the human genome or more. And usually what we mean by this is, non-enzymatic targets, things that don't have a well-defined binding site for an endogenous small molecule ligand that we can hijack. So, the opportunity space is huge in burgeoning biology, or biology that's really been off limits. And again, if we use that kind of expanded scope to include things that we're currently drugging successfully with injectable or antibody products and would like to provide a small molecule solution for, then you can start to attach some, really large numbers to these very successful antibody franchises out there as well.
Unnatural Products is developing a class of therapies known as macrocyclic peptides. Can you explain what these are?
Yeah, it's kind of right there in the name, in the sense that they're larger than average rings, so that's the macrocycle part, and then peptidic in nature in the sense that they're assembled from amino acid like stuff. And as our name might suggest, we use a lot of non-natural or non-canonical amino acids, and really kind of open up kind of the panoply of small molecule style kind of thinking. On a residue by residue basis in these peptides. So these macrocyclic peptides, they're typically around a kilodalton or two. So you're much closer to the small molecule end of the spectrum on molecular weight than antibodies, but they're able to really kind of have the best of both worlds. Sometimes they're referred to as these Goldilocks molecules because they're able to really have the distribution and pharmacokinetic properties of small molecules, namely being orally bioavailable and passively cell membrane permeable in some cases, but combined targets with the selectivity and affinity is typically only found in antibodies. So multiple single digit picomolar reported ligands out there are antagonists that are making their way through the clinic, just with really exquisite selectivity profiles. So I think they really offer kind of this best of both worlds opportunity space, where we get kind of the devil we know from small molecule in terms of being able to really kind of permeate the body and access compartments that are just off limits to antibodies, but act on biology that is really beyond the scope of traditional small molecules.
Despite the company's name, Unnatural Products, macrocycles do occur in nature. What role do they play biologically?
Yeah, I mean, macrocyclic peptides, in some ways, we view as nature's solution to hard targets when you have to care about things like passive permeability and kind of surviving out in the wild. So we've been using macrocycles as successful drugs that were found in nature for decades. So the broader class of these includes things like vancomycin in the antibiotic space, but then some of the really canonical examples are in the immunosuppressive space, like cyclosporin and FK506, which sometimes I like to point out these macrocycles are so important that their biological targets are actually named after them. So cyclosporin binds cyclophilin, and FK506 binds FKBP, FK506 binding protein. And these were not just kind of seminal tools of chemical biology that let us figure out kind of some of these crucial pathways, but they've become mainstays of immunosuppressive products for decades. So yeah, nature figured out how to do this kind of Goldilocks chemistry a while ago, and really the revolution that's happening now with Unnatural Products and others in the space is being able to really pull from nature's toolbox and apply this in a more designed and synthetic strategy.
You're using an AI engine to generate synthetic macrocyclic peptides. Is it understood what characteristics these need to have to be orally available and act like a drug?
Yeah, my co-founder and I both came out of Scott Lokey's lab at UC Santa Cruz, and Scott was really kind of a field leader in understanding how cyclosporin and some of its brethren, get away with this neat trick of being, 2 to 3 times the molecular weight of traditional small molecules, and yet maintaining that oral bioavailability and passive cell membrane permeability. And the reason we booted up Unnatural Products was there was really kind of an aha moment 2/3 of the way through our PhD, where we started using these larger, more combinatorial approaches to ask questions about simple, basic molecular properties like solubility and lipophilicity. And I think we were just very data poor when we joined Scott's lab in the early days. There was a handful of examples of these synthetic molecules in the class that were orally bioavailable or passively cell membrane permeable. And by the time we left, we were routinely discovering thousands in a single experiment. And that was really kind of the ability of us to, I think, answer that question that you just asked and say, yes, we do have a handle on the compositional and structural rules that underpin these properties we care about. And now we're going to go apply that to some, interesting and valuable biology.
Well, what does it take to engineer synthetic macrocyclic peptides, and what's the process for doing that? How much of that occurs in silico versus in a wet lab?
Yeah, we're strong believers that those two technologies are best united, and so we're the process, for ligand ID, there are a number of technologies that work quite well there. Things like phase display, mRNA display, DNA encoded libraries, all empirical approaches that can find you a macrocyclic peptide ligand very readily. And then the challenge has really been in what comes next. How do you then optimize what comes out of these platforms? And while we're starting to see some improvements on kind of dragging those platforms towards this permeable drug-like space, the challenge is really in the medicinal chemistry that comes next, because tuning these scaffolds is just a little bit more complex and a little bit more challenging than traditional small molecules. They're larger, they have more amide bonds, they don't behave really like a peptide or a small molecule. There's something in between. And so, our platform is really booted up to solve that explicit problem. Like, how can we optimize these scaffolds for the properties and selectivity profiles against the biology we care about here very rapidly and efficiently? And the way we do that is with, a strong empirical component in the lab. We do, directed parallel chemistry, kind of a design make test cycle writ large for these species, and then crucially also a strong stack of in silico tools, some of which are in vogue, like machine learning or AI we call it now, some of which are more banal but just as important, like really being able to speak macrocycle fluently in the computer, coming up with our own limb systems and these kind of things to really kind of work with this evolving molecular class that's often a round peg in square holes of kind of traditional tool stacks. So yeah, the process is, find a ligand using one of these validated approaches. Sometimes computational approaches can play a role there as well. And then, really kind of iteratively refine the properties and potency and selectivity profile of that molecule to arrive at something that is truly lead-like, drug-like, and ready for more downstream preclinical assays and testing.
Beyond undruggability, what makes a target or indication well suited for a macrocyclic peptide therapy?
Yeah, I think, we don't like to pick fights with small molecules. So I think if you can do it with a traditional small molecule, you probably should, in the sense that if you're really achieving like an MOA with a small molecule that is desirable for that target, you're not kind of, giving on some axis too much there, then that's not really a great place for a macrocycle. What that leaves is everything else, and that's a pretty big opportunity. So, I think in the oral replacement of injectable thesis, there's a number of opportunities there in the I&I space and metabolism spaces that we're currently pursuing. In the intracellular undruggable space, there's some of the high-hanging fruit and precision oncology targets have fit into that large class for a while, and that's the nature of a collaboration we have with Merck and some early work of the company as well in that space. So I think the technology and the actual molecules of interest are truly indication agnostic, but obviously within that broad panoply of potential biology, winnowing that down into like the best fit for the platform, the technology, and the time is part of the work that we do around here.
The company has raised over $35 million, but it's also had success securing funding through partnering deals. Most recently, in December, the rare disease drug developer BridgeBio exercised an option to license macrocyclic peptide candidates that you discovered. How validating is this for your approach?
I think very validating. It was really great to work with that group over at Bridge. There were some fantastic drug discovery and development experts that we were working with during that collaboration, and I think we built a lot of trust with them on our platform's ability to deliver some really truly unique and exquisite molecules. I mean, I'm excited whenever the veil comes off a little bit more, and we can share some of the details. But these things are incredibly potent. Down, it's the most potent of them are down in the femtomolar range, so beyond that of even really potent antibodies, but against a completely undrugged intracellular protein-protein interaction, and crucially with a really exquisite selectivity profile that was required for that program. So I think they've been kind to let us share some of the blinded data, at least at conferences and conversations like this. But I think the inking deals is one thing, but delivering on deals is a whole nother, right? Like there's a lot of platforms that I think promise the moon, but can't always quite deliver that landing, and so this is, I think, an important point for the company of not only can we get these partners interested in what we say we're able to do, but then we're able to deliver on it and actually put some really valuable molecules back in that partner's hand, who I think is going to be the right partner to run that down the football field to patients. So yeah, super excited about that, and I think we're cheering from the sidelines now, but rooting them on.
When the deal was first announced, it simply described it as being focused on genetic diseases and cancers with clear genetic drivers. Can you say anything about the indications or the dollar amounts?
Not yet, but yeah, hopefully more to come on that down the road.
Separately, at the start of 2024, Unnatural Products entered into a collaboration with Merck to discover macrocyclic peptides for an undisclosed oncology target. This included undisclosed upfront payments and milestones of up to $220 million. Unnatural Products is pursuing its own pipeline of therapies, but what's the partnering strategy? How do you see that balancing with the development of your own pipeline, and how important a source of funding is it?
Yeah, I think I've really enjoyed walking this tightrope between the partnering model and the wholly owned pipeline. I think once again, I think those are really well served together for a platform company. At the end of the day, on natural products, we can't be experts in every TA area, or TA. We can't build and staff for every specific biological target, and some of these things are probably better served in pharma's hands as well. So really kind of being thoughtful about kind of how we build our internal pipeline versus what makes sense to partner on early has been part of the calculus really from day zero. But then additionally, as you point out, these are sometimes called non-dilutive dollars. I don't think that's quite true. They're dilutive in maybe not the cap table, but in other ways. But they're also very enriching dollars. You learn how the partner's thinking. You get exposed to different groups and expertise around either the biology or where they see the value of the application of the technology. And I think as we're building the company, that was really crucial, valuable. And then providing an additional source of dollars that we can more directly spend on, advancing the platform kind of 2.0 components, continuing to make sure that we're innovating on that front, and then use kind of some of these more equity dollars allocated to building out that wholly owned pipeline that is, the long-term value driver for Unnatural Products and its shareholders. So I think it's been a really, successful strategy to date. I think we'll continue to do it as we move forward here, and with some really great partners in both current and future, I think, as we move forward.
Your lead experimental therapy is a treatment for obesity. This is a next generation obesity drug that has the potential to avoid some of the tolerability and compliance issues of GLP-1 therapies. While there's been a lot of excitement about GLP-1 therapies, what's been the downside?
Yeah, I mean, I think first and foremost, this is like a watershed moment. I mean, it's really exciting to see. I think the metabolism field shift from it was largely treated as the third rail of drug discovery, kind of littered with past failures, to something that is just a paradigm shift in how we approach this epidemic and disease, and also I think really kind of an exercise in kind of preventative medicine where we're starting to see all these really exciting downstream functional readouts for these, and everything from obviously cardio protective, but also neurologically protective, and a number of other areas that I think it's really exciting time, I think, to be in drug discovery working in this field. That being said, obviously no drug is perfect, and the GLP-1s and their associated double and triple agonist plays are no exception. These things mostly exert their weight loss effects through calorie restriction. It's really like pharmaceutically induced kind of starvation that ends up happening, and because of that, there's a number of issues that can come along with that. You see fantastic amounts of weight loss, but around at least 25% of that is lean muscle loss, and typically the kind of suppression of the desire to eat, and the kind of resulting nausea and some of the other AEs that come along with these things cause kind of poor patient compliance over the long run. That patients get onto these things, take them for a while, and then see this kind of miraculous weight loss effect, and then want to stop. Kind of taking these injections or soon to be orals that allow you to lose this weight, and then the weight comes back, and what comes back is often predominated by fat. So you can end up in a worse body composition than when you started the cycle, and that's something that can be really problematic, especially for elderly patients, but for all patients. And so what we're excited about are some of these other pathways that can either ameliorate that effect in combinations with the GLP-1s or potentially serve as just completely orthogonal paths to achieve similar kind of fat loss paradigms, but without the associated lean muscle loss.
You're developing an activin type 2 receptor antagonism therapy. How exactly does this work?
Yeah, so this is in the greater myostatin pathway, TGF-beta family. So this class of receptors, typically are responsible for mediating muscle growth through the ligand myostatin and associated ligands that pause that muscle growth. So if you inhibit the inhibitor by antagonizing the receptor, then you essentially promote muscle growth. But I think what's also getting worked out more and more is the role of these receptors and signaling pathways in the adipose tissue, the fat tissue as well. And so in some really pioneering work done by Novartis with their receptor antagonist antibody called bimagrumab, which was spun out into a company called Versanis, which Eli Lilly later bought for around $2 billion. They showed in a pretty seminal phase 2 study that as a monotherapy, you were able to drive 20% fat loss as measured by DEXA scan, but with 4% lean muscle gain, so muscle hypertrophy in that same period. So the overall weight loss was less than these GLP-1 products, but that's because you were literally trading fat for muscle in this kind of metabolic shuffle, if you will, and crucially, the calorie intake during that period was unrestricted. So I think it's a really fundamentally different approach to this, and it's some early hints that it might be a more durable weight loss as well. So even after discontinuation, they didn't see the rapid rebound in the way that some of these GLP-1 products do. So we're super excited about this biology. I think it's well demonstrated at this point to be pretty functional and safe in patients by bimagrumab's early performance. And then, there's, of course, a number of players getting into the space. We saw Versant and AstraZeneca Inc. Built to buy with Six Peak Bio, who's following in the space with a kind of Gen 2 antibody product, and I think a few others coming out of the woodwork here. So we'll be, to our knowledge, the first, oral species that directly mimics the antibody MOA on this pathway.
And what's the development path forward?
As fast as we can. We're eyes on the prize and getting to the clinic. I'll say that without putting my foot in my mouth, things are moving fast. And I would say of all the programs we've worked on, this one's really got legs and is moving quite quickly down the path. Hope to be in and through phase 1 with the proceeds from the next funding round. So I think we'll be. I'll give you an update when we're there.
Unnatural Products, as I mentioned earlier, has raised about $38 million to date. How far will existing funding take you, and what's the plan for raising additional capital?
Yeah, we're currently in a strong cash position, which is fortunate for us to be in, and gives us kind of some optionality. However, I think it's time to double down on building out this wholly owned pipeline. I think the business and the team is evolving to really be able to execute on that thesis. And so our plans are to raise additional capital, both through non-dilutive and equity sources over the next year, just to really allow us to scale and double down on what's important. So we'll be heading out in the new year and looking forward to what I think is going to be a pretty fun 2025.
Cameron Pye, CEO and co-founder of Unnatural Products. Cameron, thanks so much for your time today.
Thank you, Danny. Appreciate it.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.